Hello. Uh, this slide has two different topics. They don't have any connection to each other. Control by sigma factor, yes, it's part of gene regulation, but DNA repair is a topic on its own. But you're going to see more of that when you get to second year and third year. So I'm just going to be introducing DNA repair after talking about control by sigma factors. Now, sigma factor, okay, what is sigma factor? Remember, if you remember that lecture on transcription, I showed the, let me just go back to transcription. Right. In the process, okay, here, all right, you can see that the different the polymerase has been shown to give different shapes. Here you have the sigma factor with it, and then and as it goes into the process of elongation, you no longer have the sigma factor. Why? Because remember, to start off transcription, polymerase has to recognize where the promoter is, and this recognition of the promoter sequence is mostly achieved by the sigma factor molecule. Polymerase itself is not able to identify promoter sequence. So this is the job of the sigma factor. With binding of the sigma factor with polymerase allows increases the efficiency of polymerase perhaps by 1,000,000 times. You know, without it's like the eyes for polymerase to be able to start to recognize a promoter sequence and then start transcription there. And once the polymerase can start transcription, then sigma factor is no longer required, leaves that polymerase, and this sigma factor will then go on to help other sigma factors. So how is sigma factor used in gene regulation? Sigma factor binds to this region. Again, this is an example of the promoter region. I think I gave an example. This is the start point, and you count backwards, -1, -2. And when they compared about 100 genes, they found that, you know, within this, there are a lot of similarities between other genes. There are two, and here also. So this is where you call the -10 sequence. It's also called the Tata box. It's also called the Pribna box. It has different names for it. And then you have the -35 sequence, and they found that this sequence are conserved. Conserved means, you know, in a lot of genes you can actually see, maybe not necessarily TAAT, but you know, other versions, but it's just found very frequent in the -10 region. Okay, now this is the binding for sigma factor. This is the polymerase. All right. I didn't actually mention the structure of the polymerase, alpha, beta, beta prime. In a polymerase, you know how in replication you have a DNA polymerase, you have the helicase, you have the gyrase, you have the primase. So these are all different molecules that makes that works together, right? So in transcription, the RNA polymerase itself is also made up by different subunits. So you have 2 alpha subunits, you have the beta subunit, and then they have the beta prime. So alpha 2 beta beta prime, this is the enzyme that does transcription, and that is what you call as the core enzyme. I mentioned that in order for the core enzyme to recognize promoter sequences, it requires the help of the sigma factor. When sigma factor binds to the core enzyme, this is called the holoenzyme, right? Without the holoenzyme is made up of the sigma factor and the core enzyme. The core enzyme is made up of alpha 2 beta beta prime. Okay. Now, Core enzyme and holoenzyme are distributed in a very, very free. Holoenzyme stable complex. Core enzyme is inactive. Loose complex means it doesn't bind DNA that well. But with the help of sigma factor, it becomes a holoenzyme, then it becomes a very, very efficient molecule, right? So this is 5 to 1000 core enzymes. So this is just a slide to show the activities of the core enzyme, the holoenzyme. Now, when they looked at sigma factor, they found that you've got different types of sigma factors. You've got some sigma factors that is called as the general sigma factor. General sigma factor means these are the sigma factor that used but in most genes, right? It's the same sigma factor that that can be used to recognize a promoter, many different types of promoters. So the general sigma factor. But as it is, they also found that there are other specialized sigma factor. And this sigma factor, specialized sigma factor, binds or identifies specific sequences. Generalized sigma factor binds to the TAT. Look here, this is the general sigma factor. It's called the sigma 70. 70 is the molecular mass, all right, 70 kilodaltons. So sigma 70 general, it binds to the TTGACA. This is a -35 sequence. It also binds to the -10 sequence. This is found in a lot of different genes. But you have the sigma 32, a different sigma factor. That this one binds to CCC-TTGAA and CCC-GATNT. And these sequences are sequences that are located in heat shock proteins. What are heat shock proteins? Proteins that that helps the bacteria to in response to heat changes in temperature. You know, some bacteria becomes a spore. Some bacteria, becomes inactive. Why? Because these genes that are produced in response to heat are being expressed. Why are they being expressed? Because you have the sigma factor that is specific for heat shock proteins. Sigma E is also a heat shock protein. This sigma factor, sigma 32, sigma A, sigma, they don't recognize the general sequence. The general sequence is only recognized by sigma 70. Okay, so only, for example, if you need to use nitrogen, right? Here you can see here one that's nitrogen. You need to switch on the nitrogen pathway. Then the first thing you need to do is to express the sigma 54. When you have the sigma 54, then it can guide the polymerase to look for genes that are going to be useful for nitrogen consumption. Right? Without the sigma 54, then that gene is not being expressed. So you understand the concept of specific sigma factor for specific functions, right? Sigma 32 increase when temperatures increase. Sigma 54 is activated when ammonia is absent. So you need a different source of nitrogen. So these are sigma factors that control gene expression. Okay, I think, this is what we are going to see. This is a process where the gene, phage is a virus, a phage, right? The SPO1 is a type of a virus. It infects bacteria. As it infects bacteria, it switches on different genes, and this is done by having different sigma factors. For example, just if you read this, right? In Bacillus subtilis, polymerase is like that of E. coli, the polymerase. General sigma is sigma 43. That's the general. So the sigma 70 is in E. coli. Yeah? So in Bacillus, the general sigma factor is 43. Infection by SPO1 activates a different transcription pattern controlled by a set of new sigma factors. Like lambda, SP1 has early, middle, late genes. So this is the genes in the virus. SP1 is a virus. So as it enters, early phage genes have promoters that are recognized. As the virus inserts its DNA into the bacteria, the first genes, the early genes, these are categorized as genes that are required early in viral infection. These genes have promoters that mimics the bacteria's promoters. So sigma factor doesn't know that this is a viral DNA. It recognizes a promoter and says, okay, polymerase, come here, and then we'll just start transcribing. But little did they know what they are transcribing is actually viral genes. Among the viral genes that are being transcribed, among them are early gene 28 codes for a new sigma factor. So the system is as such that it tricks the bacillus sigma factor to produce, to bind to the general sigma factor, the viral sigma factor, and produce a gene, and this gene is actually another sigma factor. Now, when you have a new sigma factor, this sigma factor then can take over the polymerase and then guides the polymerase, because this sigma factor is now specific for viral genes. So then it will start. So here gene 28 codes for a new sigma factor that displaces the bacterial sigma factor. Gene 28, GP28, this is the sigma factor, coenzyme complex transcribe middle phage genes, and then this gene then produces even more genes, even more sigma factors. So middle genes 33 and 34 codes for protein that replace GP28. This is also a sigma factor, and then this sigma factor will produce even more other genes as well. Right. So in this particular example, it shows that polymerase, the activity can be controlled. Activity of polymerase can be controlled by having different sets of sigma factors. With the first sigma factor, it recognizes the general promoter. With the synthesis of GP28, it will express the middle genes. Middle genes, you get the 33 and 34, then this one will express the late genes. And so you are controlling, they're expressing different genes based on, by changing the sigma factor. Okay, I hope you can get that. This slide just tells you they've done research on it. So if there is a mutation in 3, it cannot initiate the late gene. You need to understand the organization of the viral genes. I'm not sure whether you will see it in any class, but it is a normal typical virus will have early genes, middle genes, and late genes. So this is saying that, if you don't have that, then the middle genes will not be activated. If you don't have that sigma factor, then the late genes cannot be activated. So that's why these genes are specific for the middle and late genes and so on. So it's like a cascade. 1 produces 2, 2 produces 3, 3 produces 4. So without 2, you don't get 3. Things like that. Okay. This is another example for sporulation. Have a look at it, and then it's the same thing. How the cell how sporulation occurs. You have 2 compartments. So this is the forespore, and then this is the mother cell. This is the forespore. It's called the forespore. Before it becomes a true spore, it's called a forespore. And then this is the mother cell. You'll see that this table shows the interaction between the mother cell and the forespore. Holoenzyme, the bacterial cell, produces pro-E. Then you have a sigma E is activated, displaces sigma 43. And what it does is that sigma K, then sigma 43, then sigma K is created. It transcribed by sigma E, and then sigma K is activated, then displaces sigma E, and and so on. So this again, phosphorylase activates synthesis of sigma F. Sigma F replaces sigma 43. Again, 43 is the general one, replaces it, then it becomes specific. Then it goes there, sigma F core complex transcribe early sporulation, and goes there, and then so there is an interconnection, is a cascade. Once you switch on the one gene, this will transcribe a few set of genes. This new set of genes includes a sigma factor that will transcribe even new sets of gene, and so on and so forth. All right. Okay, I'll do another slide for the repair system.